Unlike in sea urchin, where the acrosome reaction is triggered by the contact of the sperm and the egg, in mammals the acrosome reaction usually happens before the sperm makes contact with the egg. The acrosome reaction is thought to be induced by progesterone, and if you recall, the progesterone is going to increase in concentration near the oocyte because it is secreted by the cumulus cells. The mechanism by which the increased progesterone concentrations lead to the acrosome reaction is not clear, but is thought to involve, just like in sea urchin, an increase in calcium concentration. Now, the sperm that are successful at fertilizing the egg usually undergo the acrosome reaction before they come in contact with the cumulus cells. However, some evidence suggests that the acrosome reaction may also be induced by glycoproteins, which are known as zona pellucida proteins or CP proteins. And these are present, as the name suggests, in the zona pellucida of the egg. Mice express 3 CP proteins while humans have 4. Of the 4 CP proteins, the human CP2 protein has been shown to be critical for the binding of the sperm to the zona pellucida. This was shown because expressing the human CP2 protein in mouse oocytes allowed for the binding of human sperm to these mouse oocytes. But don't worry, there were no mouse-human chimeras that were created as a result of this experiment. This experiment simply showed that in humans, the binding of the sperm to the zona pellucida is mediated by CP2, because the other zona proteins were expressed in these oocytes but were unable to induce sperm binding. Once the sperm binds to the zona pellucida, it can begin to digest a channel through it to reach the cell membrane. After the sperm binds to the zona proteins and digests a path through the zona pellucida, it's finally able to fuse with the egg. As you can see in this picture, it is the side of the sperm rather than the tip that fuses with the egg cell membrane. As in the sea urchin, the acrosome reaction not only results in the release of proteases, but also in the integration of the acrosomal membrane into the sperm's plasma membrane. So this exposes proteins that were previously part of the membrane of the acrosomal vesicle, because now these proteins become localized to the plasma membrane of the sperm. In the case of the mammalian sperm, one of the proteins that is unmasked as a result of the acrosome reaction is isomo, a protein that is required for the fusion of the sperm and the egg. Izumo, by the way, is the name of a Japanese shrine dedicated to marriage, so this is where the name of this protein came from. And Izumo, which is on the sperm, binds to Juno, which is on the egg, and is shown here in blue. And the name Juno comes from the name of the Roman goddess of love and marriage. Juno and Izumo bind to each other and they recruit other proteins like CD9, which create an adhesion and fusion complex. Now, because Juno and Izumo are essential for the fusion of the sperm and the egg, mutations in either of these proteins or deletion of these proteins result in infertility. Additionally, mutations in the proteins that are recruited by the binding of Juno to Izumo, such as CD9, also block fertilization. Now that we've looked at the process of fertilization and the fusion of the sperm and egg, let's look at the processes that help to prevent polyspermy in mammalian cells. We've already looked at one of those processes, which was the binding of the sperm to the epithelial cells of the isthmus of the oviduct. Another process that helps to prevent polyspermy is the fact that in mammals, there is a slow block to polyspermy, like in sea urchin. Unlike sea urchin, however, there is no fast block to polyspermy in mammals. But the slow block functions in the same way as the one in sea urchin. It's the cortical granule reaction. The cortical granules that are located within the egg undergo exocytosis, and that happens as a result of a calcium wave that's triggered by the entry of the sperm, just like in the sea urchin. As a result of that, the cortical granules release proteases that help to remove bound sperm. In this case, in the case of mammalian cells, this is mediated by the cleavage of the CP2 proteins. So once the cortical granule reaction happens, CP2 is removed, is cleaved, and as a result, the sperm are also removed. If CP2 is mutated so that it cannot be cleaved by the proteases that are released from the cortical granules, the sperm remain bound to the egg and the rate of polyspermy increases. So that suggests that in order for this slow block of polyspermy to be effective, the CP2 has to be cleaved from the oocyte. Another slow block to polyspermy that was described very recently, so this is actually not in your textbook, is known as the zinc spark, and as the name suggests, it involves the release of zinc. This is triggered by the entry of the first sperm, which causes billions of zinc ions to be released. And these zinc ions bind to the zona pellucida. There, the purpose of these ions is basically to inhibit acrosomal enzymes. And remember that the acrosomal enzymes are going to help the sperm to digest through the zona pellucida. So these zinc ions, by inhibiting these acrosomal enzymes, help to block the passage of sperm through the zona pellucida. As we saw previously, the cortical granule reaction helps to cleave CP2 from the zona pellucida to remove any sperm that may have bound there. But the cortical granule reaction also has another very neat effect, and that is that it leads to the release of Juno from the egg cell membrane. So recall that Juno, and Juno is shown here in purple, binds to isumo. So isumo is on the sperm, Juno is on the egg. And the binding of Juno and isumo is necessary for the fusion of the sperm and the egg plasma membranes. When the cortical reaction happens, what occurs is that Juno is actually released from the membrane of the oocyte. So basically that Juno is being ejected so that no additional sperm can bind to this fertilized egg. But what's really neat is that these Juno proteins that are no longer part of the membrane actually bind to the sperm in the vicinity of the egg, and that further prevents the binding of additional sperm with this fertilized egg. As we saw in sea urchin, the activation of the egg's metabolism is triggered by the increase in calcium concentration that results from the action of the enzyme phospholipase C. And recall that this enzyme is activated after the fusion of the sperm and the egg. Now, this increase in calcium concentration allows the oocyte to resume meiosis, and it also induces DNA and protein synthesis. Just as in the sea urchin, the sperm and the egg pronuclei need to fuse with each other. And this process, as we saw, involves the organization of the microtubules by the sperm's centrosome, and then the migration of the pronuclei along these microtubules. In sea urchins, that migration takes about an hour, whereas in mammals, it takes about 12 hours. Unlike in the sea urchin, in which the oocyte has already completed meiosis before fertilization, in most mammals, the oocyte is arrested at metaphase 2. So it has to resume meiosis prior to the fusion of the pronuclei. And that's mediated by the oscillation in calcium levels. Once meiosis has been completed in the oocyte, the DNA of both the sperm and the oocyte needs to be replicated. In addition to that, the patterns of methylation that exist in the sperm and the egg have to be, for the most part, wiped clean. And this is so that the zygote has a genome that is typical of a totipotent cell. But remember that there are some imprinted genes for which those gender-specific methylation patterns remain. Once the pronuclei finish replicating their DNA and finally reach each other, they actually don't fuse. So you can see here, instead of fusing, their chromosomes condense and the zygote undergoes the first cleavage. So this means that the diploid zygote nucleus is not observed until the 2-cell stage. I just want to point out that while maternal mRNAs control less aspects of fertilization in mammals compared to other species, maternal mRNAs are present up to the 2 to 4 cell stage, and after that stage, they're degraded. The sperm contributes more than just its genetic material to the zygote, since it brings its centrosome as well as certain proteins like phospholipase C, which are essential for activating the metabolism of the egg. However, as in sea urchins, most mitochondria that are brought in with the sperm are actually actively destroyed once inside the egg. However, recent studies suggest that there may be exceptions to this. In some individuals, both maternal and paternal mitochondria have been detected, but this doesn't seem to be a widespread occurrence. So based on the current evidence, for most people, mitochondria and their associated genomes are inherited through the maternal line.